Then we studied the influence of elicitors and precursor feeding on the accumulation of polyphenolics in suspension culture. And here we concentrated mainly on the polyphenolics, although still going some more research on which or on more secondary metabolites is going on up till now. And the strategy was by pooling the whole or pooling the suspension cultures from many flasks all together, and then 10 milliliter back cell volume of the suspension was introduced into 90 ml of fresh media, supplemented of course with the same growth regulator medium, into 250 ml air laminar flasks, and then these were supplemented by the elicitors and precursors. And we used chitosan, sodium nitroprusside, methyl jasmonate, copper sulfate, and phenylalanine with the shown concentrations, with different concentrations, to study which concentration will produce better or the best activity or effect to increase the activity of the callus or increase the polyphenolics in this study. All of these, the experiment was performed on each concentration was performed on 6 times, repeated 6 times, or on 6 flasks, and the whole experiment was repeated at least twice, or it was repeated twice. Of course, here we've got about more than 120 conical flasks. It's a big experiment, I think. All the flasks were then incubated on a gyratory shaker at 120 rotation per minute and left for 15 days, where the cells or the suspension was harvested and kept for chemical and biological investigation. For the estimation of total polyphenolics, we used the Folin Catico reagent, and it was found that the calli supplemented with methyl jasmonate and phenylalanine possessed the highest content of polyphenols, or showed the highest content of polyphenol regarded in comparison with the callus and without any treatment and the cultivated plant or the wild type. However, upon studying the caffeic acid derivatives, which is a part of the polyphenols, we found that caphtoric acid and chichoric acid content was highest in the cultivated plant or the wild type plant, while caffeic acid, and these are the most, and chlorogenic acid and caffeic acid content was highest in the callus without any treatment, which indicates that the increase in polyphenolics was not due to increase in caffeic acid derivatives. So caffeic acid derivatives are not those who possess the increase. And we were interested a little bit with caffeic acid derivatives because the caffeic acid derivatives, some of the papers, a lot of researchers say that the caffeic acid derivatives are one of the most important components in this plant, and due to which the activity, sometimes they regain the activity to the caffeic acid derivatives. Then we proceeded to the in vitro biological screening. And the first step was in vitro cytotoxicity assay. And this was performed to assess the viability of normal cells. And the human peripheral blood mononuclear cells were chosen. The viability of the cells was measured using the neutral red uptake method or assay to determine the non-cytotoxic concentration of each extract. Or, in other words, the maximum safe concentration that keeps 100% of the cells viable. And then these doses, or the doses we got, were used later for all the in vitro biological studies or assays. Then we proceeded to the total anti-inflammatory activity. And the total antioxidant status was determined by the ABTS-based colorimetric method. And to compare the antioxidant activity of the different extracts, 50% inhibition of ABTS was calculated. And it was found that calli supplemented with, or the extracts of these calli supplemented with phenylalanine, methyl jasmonate, sodium nitroprusside, copper sulfate, and chitosan at the shown concentrations were the best to increase the ABTS scavenging activity. The next experiment was in vitro phagocytic activity assay. And this was performed by co-culturing of the phagocytic cells with the tested microorganisms, and neutrophils and the macrophages were used as representative cell types. Also, methylene blue was used as the indicator dye, and the test organism was Candida albicans. And the phagocytic index was calculated by dividing the number of Candida engulfed by the number of neutrophils or macrophages, while the killing ratio was calculated by dividing the number of killed Candida by the total number of ingested Candida. And it was found that, again, Calli supplemented with 2 milligram per mole phenylalanine, 10 milligram per liter methyl jasmonate, and 2 micromole copper sulfate were the best to increase the neutrophil phagocytic activity. And for the neutrophil yeast killing activity, it was found that phenylalanine, methyl jasmonate, sodium nitroprusside, and copper sulfate with the shown concentrations were the best. Regarding the macrophage phagocytosis, Calli supplemented with phenylalanine, sodium nitroprusside, copper sulfate, and chitosan with the shown concentrations were the best to increase the macrophage phagocytosis. Similarly, or nearly the same concentrations were effective as in macrophage yeast killing properties, with only difference that phenylalanine here is 3 millimole per liter, and in the previous one it was 2 millimole per liter, or 2 millimole concentration. The next experiment was the in vitro anti-inflammatory assay. And to measure or to evaluate the anti-inflammatory assay, it was measured by calculating or finding the capacity of Echinacea extract to inhibit the overproduction of nitric oxide produced as a response to lipopolysaccharides stimulated human peripheral blood mononuclear cells in patients. Also, the peripheral blood mononuclear cells proliferation was measured to assess the general immune functions at stimulation index 3. We use stimulation index 3 here. And it was found that phenylalanine, methyl jasmonate, sodium nitroprusside, and copper sulfate, chitosan, at the shown concentrations were the best to increase the proliferation at stimulation index 3. Additionally, the sodium nitric oxide scavenging activity was increased by Calli supplemented with, or the extract of the Calli supplemented with 2 millimole phenylalanine, 10 milligram per liter methyl jasmonate, 2 and 8 micromole copper sulfate, and 25 milligram per liter chitosan. Finally, the conclusions and recommendations. From all the experiments, it was clear that although the caffeic acid and chlorogenic acid was increased in the untreated Calli, however, the potency of the extracts was not affected at all, nearly. And this shows that the impact of caffeic acid and chlorogenic acid, or at least that caffeic acid and chlorogenic acid, has no effect on the aforementioned activities. Also, when we assess the cultivated plant, it was found that chichoric acid and caphteric acid are high, have the highest content, which, however, the anti-inflammatory and neutrophil phagocytic activities were nearly abolished or null. And finally, we concluded that supplementing the plant or the suspension with 10 milligram per liter methyl jasmonate, 2 millimole phenylalanine, and 2 micromole copper sulfate was the best, or were the best treatments to improve the biological activity of these Calli, or of these extracts. Finally, our recommendations. I think before there is a lot of debate. Again, I want to make sure of this, that everybody to make sure of this. There is a big debate about Echinacea, the use of Echinacea all over the world. If you search, you will find maybe hundreds or thousands of papers talking about Echinacea. Some of the people say that the activity is due to one of each, or some of the certain compounds, maybe due to the polysaccharides. Others say no, it's due to alkamides. Others say no, it's a combination. Some other papers even say that there is no activity and we should stop using Echinacea, it's useless. However, or other people or other researchers find out that loads of these are important. They are important, and they confirm this. Even the problem here that sometimes repeating the same experiment, you find that the activity is different. You find different activities. So, in summary, there is still a need for more research in Echinacea, especially on the active components and the mode of action. Also, Echinacea will be accepted as a rational drug only on the basis of more valid pharmacological and clinical data obtained from the standardized consistent preparations. Finally, I'd like to thank all the members of my lab and all the members and all the ones who contributed to this study, and thank you all. And if any questions. What method
Which method you was adopted to measure to increase the secondary metabolites in your in vitro culture?
You mean how I done the experiment exactly? Yes. Yeah. What we have done, we have established the suspension cultures, and then these were fed by the precursors, sodium nitroprusside, copper sulfate, and this stuff, and then we compared the content of the secondary metabolites or the polyphenolics, which we were interested in first, and this was compared with the naturally grown plant or the callus as a control without any treatment. Of course, as I've said in my talk, that we were concentrating on the polyphenolics in this study, but later or now we are going on with the alkamides and the other components to see which ones are the main reason for this biological activity or the increase in biological activity.
You have not adopted any kind of this HPLC systems or HPLC methods to measure the percent concentration available?
Yeah, we've used, it was shown in the graphs that we used LCMS for, LCR, okay, yeah, no worries. Okay. Yes. Yeah. Yes. No, it was not only the most promising. We studied all the extracts, in fact, with LCMS. In fact, what we have been doing, or what we were concentrating on was the polyphenolics. So we used all the the media or the what is called the developing system. It was mainly for the lipopolysaccharides, mainly. Sorry, the polyphenolics, or mainly for the polyphenolics and the caffeic acid derivatives, and even the standards we used were those for the caffeic acid derivatives. So we didn't have a whole view on the other stuff, and that's why we are now completing this work to find out the rest of the stuff. Yeah, still going on. Yeah. Yes. Yes, exactly. Yes, exactly. Okay, thank you.